Now, when we finished our discussion yesterday about cardiac muscle, we were talking about how the cells were joined together. And what did we call that jagged, zigzag junction, kind of like a stair step that joined the cells together? What did we call that? Some kind of disc. Intercalated disc. To intercalate means to join together, like a dovetail joint. To intercalate together, much like we saw with the Reedy apparatus in our skin joining together. And within our intercalated disc, we had two junctional complexes of proteins, one for strength and one for communication. The one for strength, like Velcro, were the desmosomes. And the one for communication that electrically coupled the cardiac muscle cells that were adjacent together were the gap junctions, these little tunnels that allow the cytoplasm of one cell to be continuous with the cytoplasm of the next. Now, when we put this together in the context of our cells, all our cardiac muscle cells of the atria and all our cardiac muscle cells of the ventricles are joined together with these gap junctions. So when a neighboring cell undergoes our action potential, sodium ions come in, changes our membrane potential. The sodium ions can also go through the gap junction to change the membrane potential of the neighboring cell. And when that happens, we get the release of calcium from the sarcoplasmic reticulum. Calcium binds to what? Troponin. Troponin changes shape and pulls what out of the way? Tropomyosin. It's the same thing. The same contraction mechanisms in skeletal muscle still apply to cardiac muscle. The only differences between the muscle are skeletal is long and cylindrical, cardiac is branched, and cardiac muscle has the intercalated discs that have the gap junctions that functionally couple them together into that syncytium. It's kind of a big new word. Syncytium, one functional unit comprised of many interdependent parts. But that's our cardiac muscle. Now, smooth muscle is the most different. We call this smooth muscle because it lacks striations. Now, do not fall into this trap. And in fact, we've got this coming up in the next couple of slides, so I'm going to wait till we get to the next slide before I go there. But smooth muscle, we find this associated with a lot of our internal organs, our viscera. Visceral really refers to our internal organs. So we find that a ton of smooth muscle is invested in the walls of our digestive tract. Your stomach is almost entirely smooth muscle arranged in 3 or 4 different arrangements of bands and belts and layers of smooth muscle. Your intestines have a ton of smooth muscle. Smooth muscle around your airways. Smooth muscle around many of your blood vessels, especially your arteries. And much like cardiac muscle, you can't control the smooth muscle. It is completely involuntary. Skeletal muscle is voluntary. Sometimes we can subconsciously control it, but it's involuntary in the sense that we can stop it. Our diaphragm, right now it's subconsciously controlled, but we can hold our breath. Smooth muscle, cardiac muscle, you have no control over whatsoever. Now here for the first time also, we're getting away from our cylindrical cells like skeletal, our cylindrical branch like cardiac. Now we have a completely different shape. And the shape of our smooth muscle are said to be spindle-like or fusiform. 2 terms that you rarely will ever use in our modern society. Basically, these shapes are rounded in the middle and tapered on both ends. Have you ever seen a spindle? Do you even know what a spindle is? We got to go back to the fairy tales, right? Was it Sleeping Beauty? Who pricked their finger on the spinning wheel? Was that Sleeping Beauty? She pricked her finger on the spindle of the spinning wheel. So as the wheel spins, that wool and that thread winds around that stick. And as that winds around it, it's usually fat in the middle and tapered on either end, much like that. That's where that word came from. I don't know that I've ever seen a real one in action, but that's where we get that from. I like to call that a stretched out football. If you could take a football at each end and just stretch it out, that's what it's going to look like. So as long as you have this shape in mind, that's the shape of smooth muscle. No longer cables, no longer cords. But this is also going to present a problem in the sense that smooth muscle is not going to have these things we traditionally call sarcomeres. But some of the components we're going to have inside our smooth muscle cell. First of all, to maintain that elongated football shape, we're going to have cytoskeletal proteins that are going to help us maintain that shape when the muscle's relaxed. And this is the first time we've really used intermediate filaments for much, but we're going to use intermediates here to maintain the shape. But we're also going to have these features that you can see histologically. We're going to have these dense plaques and dense bodies. These are collections of proteins. If they're on the plasma membrane, we call them plaques. If they're internal in the cytoplasm, we're going to call them bodies. These are anchoring points for our contractile proteins. The dense bodies and dense plaques, they're anchoring points for our contractile proteins. But if you look at this, you are not in any way, shape, or form, there's no sarcomeres to speak of. No striations. Because at each one of these little pucker points all over the surface of the cell and internal to the cell, this thing's arranged in three-dimensional patterns, not just two-dimensional cables. Or maybe two-dimensional sheets like in cardiac muscle. So this is going to present somewhat of a problem to our former understanding of how cells contract, because already we see things organized in a completely different manner. So let's look a little bit more closely at some of these dense plaques and dense bodies to get a better appreciation for how our proteins are organized inside our smooth muscle cell. So if we take a look, let's say maybe right in this region, and we look at some of these are going to be our dense bodies inside the cell. Notice what we have associated with the dense bodies. Those are our thin actin filaments. Notice between the thin actin filaments we have thick myosin. That's sounding kind of familiar, isn't it? And even though we don't have anything that we can really describe as a sarcomere and striations, notice that our dense plaques and dense bodies are serving the same function as our Z-line in our sarcomeric muscle. The Z-line that you would see in skeletal, the Z-line that you would see in cardiac, we don't have those. We've replaced them with dense bodies and dense plaques. And as we instruct this muscle to contract and we slide the actin along the myosin, just like we're going to do with our cross-bridge cycle, it's going to move the dense bodies and dense plaques closer to each other. So in a sense, these are very similar. We're going to say they're analogous. They're not synonymous. They're not exactly the same. But our position between dense bodies and dense plaques are analogous to our sarcomeres. We simply don't call them that. But I want you to understand that even though we don't have sarcomeres, we absolutely must have thin actin and thick myosin. Without those two proteins, we will not contract. Don't equate sarcomeres and actin and myosin together. They're not identical. So we have the actin and myosin. We even have tropomyosin. But we don't have any troponin. Okay, now we got a problem, right? Because we had to have troponin to move tropomyosin out of the way so we could get a cross-bridge cycle. Well, I'm going to go ahead and give you a little extra piece of information here. Tropomyosin is not blocking the myosin binding sites on actin. Tropomyosin in smooth muscle is simply a structural protein. It's not regulating anything in smooth muscle. So as far as contraction relaxation goes, tropomyosin may as well not even be there. So you can already tell our sort of excitation coupling is going to be a little different than we had in our striated muscle, skeletal and cardiac. The other thing that's different from other two types of muscle, in smooth muscle, we don't have a sarcoplasmic reticulum. Okay, well, we're still going to need calcium, right? Yeah. But we don't have troponin that binds calcium, right? We're going to have other stuff. But we still need calcium. We still need to control the release of calcium. We still need to be able to pump calcium back in. So instead of having this big structure called a sarcoplasmic reticulum, we have a lot of these little bubbles that sit just underneath the plasma membrane, just underneath the sarcolemma. And they do the same thing as the sarcoplasmic reticulum. Instead of having one sarcoplasmic reticulum, we have hundreds and thousands of these little bubbles called caveolae, subplasmalemmal vesicles they call them. I just like little bubbles. And they store and control the release of calcium. Now this is where it's kind of similar to what we've seen before. A nerve is going to signal the cell. Neurotransmitters are going to be released. We're going to repropagate an action potential on the surface of the cell. Sounds the same so far, right? That action potential is going to hit a calcium voltage-gated release channel. In our skeletal and cardiac, that was in the T-tubules associated with the terminal cisternae. In smooth muscle, the calcium release channels are associated with the caveolae. And when the action potential causes those calcium voltage-gated release channels to open, calcium floods into the cytoplasm. Exactly the same. Calcium is still going to trigger and couple the excitation with the cross-bridge cycling. We just do it a slightly different way with the caveolae. But this is where things are, again, going to be different because calcium can't bind to troponin when it's not there. In smooth muscle, calcium binds to a protein called calmodulin. This is where we're going to stack up our dominoes and knock the first one over, and then the rest are going to knock each other over until we get a cross-bridge cycle. Calcium is going to be that finger that knocks over the first domino. The first domino is calmodulin. When calcium binds to calmodulin, that activates calmodulin. Calmodulin, its job is to now go activate another enzyme. The enzyme that activated calmodulin turns on is myosin light chain kinase. Now, let's break down this name. What protein are we going to use this enzyme to modify? What's the first word? Myosin. Okay, that's easy. And we've used this type of enzyme quite a bit. What does a kinase do? What does a kinase enzyme do? It adds a phosphate. So we know that this enzyme is going to add a phosphate to myosin. But here's the other part, light chain. You remember our myosin molecule? Our arm? There's another division in which we can describe this molecule. We can break this molecule down into the heavy and light chains. The light chain, or the tail, is from your shoulder to your elbow. From your elbow toward the end, that's referred to as the heavy chain. So myosin light chain kinase adds a phosphate where? To the myosin light chain, or the myosin tail. That's going to be a way that we can activate myosin. Now, for right now, just hang on to the fact that myosin light chain kinase activates myosin. But you do realize that if you activate something, you also have to have ability to turn it off. If you turn it on, you got to be able to turn it off. And so we need an enzyme that can turn it off by removing a phosphate. So essentially we have these two enzymes. When calcium is bound to calmodulin, it goes and it tells myosin light chain kinase, hey, go turn on myosin. Go take this key, the phosphate, stick it into myosin, turn it on. That's your job. And when the calcium goes away and calmodulin stops telling the myosin light chain kinase to turn on myosin, that's the signal for myosin light chain phosphatase to go take the key out, the phosphate, and turn myosin off. That's part of our contraction, adding the phosphate, turning it on, and then relaxation, removing the phosphate and turning it off. Do we understand the difference between the kinase and the phosphatase and where the myosin light chain is? Again, we're just building the pieces. We're going to get to the big picture in just a second. Are we good so far with this? Anybody lost? Yes, ma'am. The light chain is the tail. It's going to be this upper part of our arm for our myosin model. Okay? The heavy chain is from the elbow to the end. All right, so now let's put this all together, much like we did with our striated muscle. Here we've got our nerve signaling detected by the receptors on our sarcolemma. We repropagate the action potential that goes all over the cell, and it's going to open those calcium voltage-gated release channels, and calcium is going to leave the caveolae. Calcium leaves the caveolae, and once it's inside the cell, it can bind to calmodulin, much like in striated muscle, calcium bound to troponin. So that's the first domino to fall. Calmodulin gets turned on. So now calmodulin goes and turns on myosin light chain kinase. Now here we have myosin light chain kinase being activated by calmodulin with calcium bound to it. Myosin light chain kinase, once activated by calmodulin, adds a phosphate to the myosin light chain or the myosin tail, leading to, what does that next part say? The unfolding of myosin. I want you to underline, highlight, circle, unfolding. Because this is a key concept in understanding smooth muscle contraction and relaxation. In our striated muscle, what prevents myosin from binding to actin? In our striated muscle, what prevents myosin from binding to actin? I think I heard it. Tropomyosin. Myosin can bind to actin. It's got tropomyosin in the way. Well, remember we said tropomyosin's not in the way here. The myosin binding site is wide open on actin. So what keeps myosin from binding to actin when the muscle is relaxed? And it's the fact that the myosin molecule, when it does not have a phosphate on its light chain, that myosin head that has ADP and phosphate in it, that myosin head that's ready to bind to actin, without a phosphate on the light chain, the tail here, like my shirt, everybody look. That myosin that is preset, ready to bind to actin, without a phosphate here, the head binds back to its own tail. It's folded up. It can't bind to actin. And that's all because that phosphate isn't present on the light chain. So here is our relaxed, smooth muscle myosin. See how it's all kind of folded and twisted back on itself? But whenever calcium comes in, activating calmodulin, that then activates myosin light chain kinase, that phosphorylation event happens. It happens on the myosin tail. And when that phosphate is added, myosin springs out. It unfolds. You know, like that windshield thing you put in your car, you fold it up and you kind of touch it, it pops out. Well, myosin pops out, unfolds, and arranges itself into a thick filament. Now we can start having our cross-bridge cycles. As soon as myosin unfolds, you start having cross-bridge cycles. As long as ATP is present. And the cross-bridge cycle in smooth muscle is identical to the cross-bridge cycle in skeletal and cardiac. Right? So you unfold myosin, what do you get? What is that called? Cross-bridge. Make the sound. Phosphate. What's this? Power stroke. Make the sound. ADP. We've got to bring a new ATP in, right? Reset, do it again. It's the same thing. The same thing in all three muscle types. It's just getting to the point that we can make a cross bridge. That's the different point in smooth muscle. And it involves calmodulin, myosin light chain kinase, and that phosphate added to the tail to unfold myosin. Now, once we start our cross-bridge cycles, unlike skeletal muscle, these long cables that shorten the muscle in this one plane, it's a little bit more like cardiac muscle that's arranged into sheets and layers into this three-dimensional organ called the heart that compresses in on itself. When all of these thick myosin slide to thin actin, we're going to compress that cell in a three-dimensional manner. Now, have you seen, it's about Christmas time, and they usually have this toy close to the checkout place in Walmart. Have you seen that ball, that plastic multicolored ball, that when you pull on it, it just gets ginormous? And then when you let it go, it collapses back down on itself into just a ball? Imagine a smooth muscle cell is that ball in the enlarged state, and the contraction of the smooth muscle compressing down three-dimensionally. Because that's what happens. When all these dense bodies and dense plaques shorten and pull at each other, you get that three-dimensional compressing of the whole cell. Now, when you join these cells together into sheets and layers and belts, you're going to accomplish a lot of work in the digestive system, respiratory system, and circulatory system. Now, here's an illustration. If you look in your book, you're not going to find this illustration because I edited it in Photoshop. Because in your book, they did not have a caveolae, this little bubble underneath the plasma membrane. They had a sarcoplasmic reticulum out there. What's wrong with that picture? There's no sarcoplasmic reticulum. What are you people, crazy? So again, make sure you use your critical skills, your critical thinking skills when you're reading books or reading papers or looking up anything on the internet, whether it looks like it's legit or not. Because you know there's not a sarcoplasmic reticulum, and we got that sucker out and we put a caveolae in. But there's our caveolae. Calcium comes out, binds and activates calmodulin, domino 1. Calmodulin activates myosin light chain kinase, domino 2. Pull a phosphate off of ATP. I don't like them putting the phosphate on the head because it goes on the tail. I just couldn't edit that out as easily. So myosin light chain kinase. They're not really showing the unfolding, which is regretful. That's why I had that other picture in. I put that in at the last minute. You probably noticed that if you downloaded the slides. But I couldn't fix that any better. And then you go through your cross-bridge cycle that looks the same. All right? And to relax the muscle. This just shows you a relaxed state and a condensed state. Pretty much the same, just takes up much less volume for our smooth muscle cell. But our relaxation is going to involve nerve, not signaling, calcium pumps returning calcium to the sarcoplasmic reticulum, which activates myosin light chain phosphatase. Removes the phosphate from the light chain, so in that last reset cycle for myosin, instead of binding back to actin, it binds back to its own light chain, folds up, and you store the muscle in a relaxed state until you do it again. Do you have a question?
Yes.
Okay.
I have a question.
Yeah.
What did you say cessation was?
Stopping.
Oh, stop.
Cessation, cease and desist, stop. You stop stimulating. The nerve just stops. Now, when we look at these steps to go through contracting a smooth muscle, and we look at those versus the steps for skeletal and cardiac, there's more steps here, more enzymes. And the way those enzymes get together are just random collisions until something fits. So can you begin to see that it's going to take longer to contract a smooth muscle than it would be a skeletal muscle? Is that going to be a problem? Is there ever an emergency situation in your body where you absolutely have to have your smooth muscles acting instantaneously? Give me a for instance.
What do you mean instantaneous?
Okay, when you touch a hot stove, your skeletal muscles act instantly.
Yeah.
Is there ever any kind of emergency situation? A bear's chasing you. Is smooth muscle going to be involved in you getting away from this bear? No. No. So, since it's not involved in emergency stuff, it doesn't have to be fast. It can be slower, and we're perfectly good with that. Because that latent period before you actually get a contraction takes longer. But is that a disadvantage? Does that mean this muscle is not really worth much? We'd much rather have skeletal or something else that works faster. No. Because smooth muscle works a lot longer than your skeletal muscle. Is there ever a period where your digestive tract isn't working? No. In fact, you may say, well, when I'm asleep. No, when you're sleeping, your digestive tract goes into high gear. That's why if you want to begin to lose weight, do not eat within 3 hours before you go to sleep at night. Because at night, your digestive tract is more efficient than any other time of your 24-hour period. So this thing's working all the time. So smooth muscle is slower. It's like the tortoise and the hare. It's slower, but it's going to keep going when the skeletal muscle poops out. Because your smooth muscle is fatigue resistant. We're not going for speed. We're not going for power. We're just going for steady. Steady. So it's a big advantage for our smooth muscle and the performance of its tasks. Now, the last thing we're going to cover here as far as smooth muscle goes is how smooth muscle contraction is regulated. We're either going to see regulation by each individual cell being innervated by a motor neuron, or sheets and layers of smooth muscle cells being controlled by only one. And so the first type is called multi-unit. Every cell gets instruction from a nerve. And much like we saw with our skeletal muscle, we've got motor units that can be activated depending on how many nerves we have to use to get the work done. So erector pili muscle, did you realize erector pili was smooth muscle, right? That's our goosebump muscle. The muscles that control the iris of your eye. What is the iris of your eye? What does that control? Your pupil. We also have little muscles that are attached to the lens of your eye called the ciliary muscle. That's smooth muscle. And the contraction state and the tone of that muscle controls the shape of your lens. You know, the lens in your eye isn't a solid marble. It's pliable. It's flexible. And the contracted state of the ciliary muscle, that dictates your focal point. And if you want to activate and exercise your ciliary muscle, put your finger up, focus on your finger, and then move it away and focus on the wall. Then focus back on your finger, focus on the wall. As you get older, that's harder to do, so I'm not going to do it anymore. I'll be tired. No. But that rapid change is the ciliary muscle controlled by multi-unit smooth muscle fibers. Now, we can do that for fairly small muscles. When we get into our digestive tract, yeah, this is where we're going to have whole sheets or bands and belts of smooth muscle that are all tied together and they work as a unit. Again, this is another type of syncytium, like we saw with cardiac muscle. And what junction did we have in cardiac muscle that made it a syncytium? What did we call that thing? Little tunnels? Gap junctions. Guess what? In our single unit, it's also called unitary smooth muscle. These are tied together with gap junctions. So when one undergoes an action potential, they all undergo an action potential. And even though we don't have individual axon terminals impacting on these cells, we have individual axons that run through the entire layer. And all along the length of the axon, we have these enlarged, swollen parts of our axon that are called varicosities. And it's in these varicosities where we can release neurotransmitter. And so as this neuron runs through the muscle, and it has these number of varicosities, it can stimulate the muscle to contract as it passes through. So the term for this type of signaling is called signaling en passant in French, or in passing, signaling in passing. But basically all you got to do is stimulate one, and the rest, they're going to contract as well. Because as your stomach, as your intestines are undergoing peristalsis, you don't have to have a lot of fidelity for that. You got to have a little bit more power, and it's going to be longer and sustained. Because in your intestines, you have these bands that are going to take the lumen of your intestine and make it smaller. A lot of times along the length of your intestinal tract, you have these bands that are called sphincters. You'll get to those in 239 in the digestive system. But in addition to having these sphincters, these bands in your intestines, you also have ribbons that run down the length. And so when these ribbons of smooth muscle contract, it takes that length of intestine and shortens it. It squishes it together. And then you can have another band that compresses, whereas the belt relaxes and it moves forward. Have you ever had sweatpants and the string get lost up in your sweatpants? That's the most frustrating thing in the world, right? So what do? Well, you feel where the little aglet is. Did you know that's what you call the end of the string? You feel where the aglet is and you pinch it. And then with your other hand, you bunch up the material until you can feel the aglet with your other fingers. You grab it, and then you pull the fabric down in the other direction. And you keep doing that until you get the aglet to the hole and you can pull it out, right? That's exactly how peristalsis works in your intestines using smooth muscle, either as belts or as ribbons. To compress, scrunch, compress, extend, compress, and that's how your food, your digested material, is moved down your intestinal tract. But again, is there ever an emergency in how fast you digest your Big Mac? No. That can take time, and that's why smooth muscle has plenty of time to do its job. All right. Any questions? Smooth muscle contraction, smooth muscle signaling from nerves. Because here's a point where we're going to stop talking about muscle and we're going to start talking about nerves. The nervous system. We're going to introduce this and then we'll do some more next week.